DNA replication occurs in 3 steps: unwinding phase, pairing, joining, unwinding. The enzyme DNA helicase unwinds and unwinding in a zipping. The double helix. When the double helix is unzipping, hydrogen bonds are broken, which leaves a single strand of DNA. The single-stranded binding proteins associate when DNA strands keep them separated during the process.
As it unwinds, RNA primase adds a piece of RNA, or RNA primer, on each DNA strand. This is the first step in unwinding, or the first step of DNA replication. Like, you have the parent strand, and then they're separated into different strands, and then finally replicated on the fourth, or the second. So, in base pairing, DNA replication, or DNA polymerase, selects the different nucleotides to the different DNA strands. The nucleotides are added to the 3 end of the DNA polymerase, and polymerase continues to add nucleotides to the 3rd end.
The templates make identical copies of the parental strand of DNA. 2 strands have different replications.
One is elongated, the leading strand. The other elongates away from the replication split. The lagging strand synthesizes discontinuously, called Okazi fragments, by the DNA polymerase in the 3rd to 5 direction.
The fragments are later connected. DNA ligase in eukaryotes. Each Okazaki fragment is about 100 to 200 nucleotides long.
This is the second step in DNA replication. It's base pairing, and this is the leading strand, which goes into the DNA helicase. It's the enzyme that helps make the other strand that's replicated, or that's the parental strand, and then it's separated. This is the lagging strand, and these are the Okazi fragments. And then, once they finally end, it's when DNA ligase helps connect the Okazi fragments.
In joining, the leading strand is synthesized continuously, and eukaryotic DNA replication is in multiple areas.
When DNA polymerase comes to RNA primer, and DNA removes the primer and fills the place with nucleotides. When the RNA is replaced, DNA ligase links it in 2 sections. And then this is the 3rd and final step in DNA replication. In eukaryotic cells, it's inside of the nucleus, and because eukaryotic cells are normally a lot bigger than prokaryotic cells, they're a lot longer, so they occur in multiple areas. And then the prokaryotic cells are a lot smaller, and they are not in the nucleus, so it only occurs in one place. And that's the 3rd and final step of DNA replication. The end.